Hello, you're watching Armando Hasurungan, Biology and Medicine videos. Please make sure to subscribe, join the forum and group. For the latest videos, visit Facebook Armando Hasurungan, like, ask questions, answer questions, and please post some interesting things, including your artwork. And you can change the settings to HD or original for better graphics, so you can look at the videos better. Now, this video is on enzyme and how it controls metabolism. Metabolism is a big thing in our body systems. In biochemistry, it is the synthesis and degradation of various macromolecules or molecules to provide us with energy, to store energy, and to use energy. To live, essentially. And this energy we get from food. And there are many metabolic pathways in the body, such as there's glycolysis and there's gluconeogenesis. And you can watch these on my human metabolism map video. Anyways, they're of course all interconnected in some way, these metabolic pathways. But what controls them? What helps stimulate one pathway while stopping the other? What structures or features or mechanisms do our bodies possess that help regulate metabolism? Now, metabolism is integrated and regulated within cells, between cells in organs, and between organs. So what type of metabolic controls are there for this? This video will look at how enzymes specifically, how they play a key role in metabolic control. Now, ACE is the most important enzyme control for metabolism. ACE stands for allosteric, covalent, and enzyme itself. So what is the difference between them? Well, an enzyme typically has an active site for the substrate, and actually may contain an allosteric site, a different site for a ligand, a special type of molecule that will change its structure. So for example, in an allosteric form, some molecule or ligand will bind to the allosteric site of the enzyme, and this will cause the enzyme to change its structure, which then causes it to stop the enzyme's function or to promote its function, to activate it or to inactivate it. It depends. Now, an enzyme which has been covalently bonded is different. In this case, an actual chemical group. A chemical group has bonded to the enzyme. An example is a phosphate group binding to the enzyme, which will affect the enzyme's activity. Now, other metabolic controls include SEC. Now, SEC is for specialization of organs, the enzyme levels, also product levels, and compartmentalization, such as inside the mitochondria, the peroxisome, lysosome, special organelles. Compartmentalization. So let's have a closer look at this in a bit more detail. Okay, so here is a membrane of a cell, and here's a receptor. Now, what can stimulate an enzyme within the cell, in the nucleus? What can? Well, a ligand or hormone binding to the receptor can. Or, if the molecule is lipid soluble, it can pass through the membrane. And these molecules or hormones can stimulate the transcription, the making of enzymatic genes to make enzymes from the nucleus. So the gene makes mRNA, and this mRNA is for enzyme, to make enzymes. And they will go out of the nucleus and they will get transcribed by the ribosome to make proteins, which will then form a specific enzyme. So this protein will then form a specific enzyme once translated. Now, this enzyme has a number of fates depending on what the hormone or the body wants at that particular time, and will affect the enzyme's activity in some way. If the enzyme is not needed or levels are too high, it will be degraded by the ubiquitin proteasome system. It will be destroyed. Alternatively, enzymes obviously can do its normal function by converting a substrate to a product. And once it's made the product, it can go back to the normal enzyme. Or, as we mentioned, a ligand or molecule can bind to the allosteric site of the enzyme, changing the structure and affecting the rate and function of that particular enzyme. And obviously, if you remove the ligand, it will go back to its original way. An enzyme can also be covalently bonded to, again, change its structure, which will affect it, perhaps making the enzyme inactive or active. An example is covalently attached phosphate group, such as in glycogen phosphorylase or glycogen synthase, which will affect the activity in some way. Or also a regulatory protein. A regulatory protein can bind to the enzyme, which will essentially regulate its function, promoting it or some special regulating function. Yeah. So here you can see an enzyme bounded to a regulatory protein. Now, this is just the basic fates of an enzyme. But when do actually enzymes need to control metabolism, and what's an example of such a situation? Well, a good one, an important example is glycolysis and gluconeogenesis. Now, which is important for regulating blood glucose levels, making ATP or acetyl-CoA for fatty acid synthesis, etc. Now, this pathway, glycolysis and gluconeogenesis, is regulated carefully by enzymes, and the enzyme's activity itself can be decreased or increased, stimulated or inhibited, depending on a number of things. These things include the products and the substrates. The next video, we will look at the regulation of glycolysis and gluconeogenesis, the key enzymes, molecules, and products that play a key role in regulating this process, stimulating it or increasing it in different stages of glycolysis and gluconeogenesis. And notably, a key player is fructose 2,6-bisphosphate, which you might have heard of. So that's the next video. I hope that was good. Please like, comment, and subscribe. Thank you.